All right, I'll start now. So welcome everyone. I'm Serene Markosian. I lead the Assay Guidance Manual Translational Science Resources Program here at the National Center for Advancing Translational Sciences at the National Institutes of Health. We're really excited to have you here today. So this is our third webinar of the 2023 AGM Preclinical Translational Science webinar series. We're very excited to have Dr. Jay Bradner join us today. So Dr. Bradner served as president of the Novartis Institute for Biomedical Research, NIBR, from January 2015 to November 2022. He led global internal and external research and early development covering 8 disease areas, including hematology, oncology, immunology, neuroscience, and others, and 5 therapeutic modalities, including chemistry and chemical biology, biotherapeutics, gene therapy, and others. In his 7 years of leadership, NIBR innovated more than 80 development candidates, filed 75 INDs, and reported out 90 positive proof of concept studies in human clinical investigation. He also led a research initiative which was focused on molecular glues for intractable protein targets. Prior to leading NIBR, Dr. Bradner served on the research faculty of Harvard Medical School and as an attending physician in stem cell transplantation at the Dana-Farber Cancer Institute, where he studied BET bromodomain proteins and their function in gene control, innovating chemical probes, and investigational drugs to study and treat cancer. He also pioneered a first all-chemical strategy for targeted protein degradation, which has served as a discovery platform for many academic investigators and biopharmaceutical companies. He's a co-founder of 5 biotechnology companies and has co-authored more than 250 scientific publications and over 50 United States patent applications. He's a graduate of Harvard College and the University of Chicago Medical School. He completed his residency in medicine at Brigham and Women's Hospital, fellowship in medical oncology and hematology at the Dana-Farber Cancer Institute, and was a postdoctoral training and has a postdoctoral training in chemistry and chemical biology at Harvard University under the mentorship of Professor Stuart Schreiber. His talk today is entitled Gene Control Therapeutics. And on behalf of everyone here at ANCADS, all of my colleagues and everyone attending today, I would like to say we're really excited to have you here, Jay, to join us today at this webinar. And before I'll turn it over to you, I'd like to ask everyone attending here to place your questions in the Q&A box, and we will address those questions at the end of the talk. All right, so turn it to you, Jay. Thank you so much for joining us again.
Boy, Serene, thank you so much for the wonderful introduction. I really appreciate the chance to visit with all of you. And I'm very sorry that we're not all together, but hopefully we'll have that chance to return to that way of life very soon. I will now attempt to share my screen. I sincerely hope this works. Serene, are you able to see the slides okay?
Yes, I see the slides.
Okay, all right, thank you. Well, I really warmly appreciate the invitation, and I wanted to share some reflections from my work in ligand discovery as an academic scientist, and also in this wonderful 7-year chapter of therapeutic discovery at the Novartis Institutes for Biomedical Research. My interest historically has been in gene control medicines, chemical tools that can help us understand the way genes are regulated, and also to consider these molecules in translational model systems for mechanistic study, perhaps to pharmacologically validate a target and create an opportunity for therapeutic intervention. And I'm going to probably not talk about everything in this image right here. And Serene and colleagues invited some reflections on assays developed or utilized in the creation of gene control medicine. So I'll try to highlight that as we go. As this is a non-commercial interaction, I just want to disclose these potential conflicts of interest, most of which have been already shared with you today by Serene. All right, so gene control, it turns out to be vital for really every facet of human biology, from developmental biology to the challenges of disease. I trained as a cancer doctor before I became interested in ligand discovery. And so much of what I'll share today is framed in the context of cancer, but I do hope and expect that the insights for the study of gene control in cancer and the tools and probes and leads created herein will be equally transferable to inflammatory diseases and other serious medical diseases. Rick Young and I had the pleasure of writing this manuscript with Dinesh when we were collaborating so closely together, recognizing that cancer has sort of a transcriptional addiction, that for sure the convergence of all these signaling pathways that drive mitogen-independent cell growth changes nuclear chromatin, creating an anabolic metabolism, creating an anabolic mitochondrial state, indeed mitogenesis, as well as ribosomal genesis. And there's a program for cell duplication that becomes co-opted by any number of upstream signaling pathways, but it tends to converge on a small number of canonical growth states in the nucleus that are genetically encoded by transcription factors. And so from a therapeutic standpoint, it's quite exciting to imagine small molecules that would pharmacologically offer control, exogenous control to growth states, establishing perhaps even a therapeutic window between tissues addicted to growth genetically, like cancers, versus their healthy counterparts. And this field is very strong and very broad. And though I and colleagues at Novartis have done some really interesting work on chromatin modifying enzymes and epigenetic readers, I really wanted to focus this interaction around the highest hanging fruit, the most exciting, but also the most difficult targets in the space, the transcription factors. Transcription factors are proteins that classically associate with DNA through sequence-specific molecular recognition, and then have a multi-domain structure so as to recruit repressive complexes to silence genes or activating sequences in order to activate genes. And the transcription factors turn out to be fantastic targets for any number of diseases. And here I show a slide that highlights that. Where available, small molecules that target transcription factors are some of our best therapeutics. Think of dexamethasone, perhaps the first molecule to offer an overall survival advantage with SARS-CoV-2, limiting the inflammatory storm subsequent to systemic viral infection. Tamoxifen and AR modulators for prostate cancer. These are very important paradigmatic medicines for the treatment of breast and prostate cancer. And beyond cancer, the study of non-alcoholic steatohepatitis, the FXR transcription factor emerges as a compelling target. And these are all really important medicines, either broadly utilized or in development today. And so when we can make molecules that inhibit or activate transcription factors, they can be amazing medicines. The trick is that these proteins evolved to bind small organic molecules, be they bile acids with FXR, or natural sex hormones for these others, or steroid molecules like the glucocorticoids with GR. And so it's not a leap of too much imagination to create small molecules that could outcompete a natural ligand. But the reality is, beyond these four examples and the broader family of nuclear hormone receptors, most transcription factors don't have hydrophobic pockets where one might position of drug-like and bioactive organic molecule. And that's a great challenge to us. This was really driven home for me as I was training, and the genome sequencing of so many different cancers was being contributed. And sure enough, recurrent genetic alteration, somatic alteration of gene control factors was a common feature of cancers. And so if all we are to do, and it can't be, but if all we are to do is to enumerate small molecule modulators of all the somatically activated gene products of cancer, we're just not close to completing that job. And the transcription factors are one of the greatest unmet needs. As I was reemerging in the laboratory at the end of my training as a hematologist, I encountered this incredible paper from Jim Darnell that really inspired me to do the work that I continue to do today. And that was sort of to lay down the gauntlet from a career pioneer in the study of gene control to say, the stumbling block to creating inhibitors for these proteins is to get beyond the conceptual challenge of disrupting protein-protein interaction. And, pharmaceutical drug discovery tends to really aggressively collect the low-hanging fruit. And for sure, transcription factors are the high-hanging fruit, and they've received comparably little attention. And so I went back in the lab. This is a file photo from that time, a lot more hair, better fitness, because to discover small molecules for transcription factors meant that we wouldn't be able to rely on assays that were enzymatic measurements, perhaps. Without pockets, what biochemistry would we follow? That these proteins, by and large, function by protein-protein association, like MYC and MAX binding through an HLH leucine zipper at EBOX DNA to turn on the 5 to 15% of genes in the genome governing growth. And so one approach would be to not worry so much about function and just find molecules that bind, and then among the binders, look for inhibitors or engender inhibition or other biophysical properties through follow-up chemistry. And at this time, DNA microarrays were all the rage for monitoring transcriptional profiles and ultimately somatic alterations in the genome. And so we had one of these arrayers in Stuart's lab, and Angela Kohler and I created these small molecule microarrays as well as methodologies to apply crude cellular lysates to witness binding events to proteins and small molecules in a miniaturized format. Now, so many years later, I'm still working on assay methods to bind transcription factors. And I will not present this work today, but you must read this fantastic piece with Stu Schreiber, Fred Zechary, Corinne Briner, and so many friends at NIBR and the Broad Institute, where we undertook to elaborate 3.7 million novel molecules on DNA as a DNA encoded library, not in array format as on the left, but to bring some of the complexity and the stereochemistry of natural products to these large collections. These are so-called diversity-oriented synthesis molecules. Imagine 61 skeletons and almost 300 amine capping building blocks, 206 boronic acids, and library synthesis with building blocks as shown here that have 8, and even 12 isomeric forms. And I hope that this resource, now established for its open access, leads to the discovery of small molecule binders to intractable protein targets. So I'm going to tell two quick stories and then focus in a little bit on my current thread of investigation to target transcription factors, even those that have a lot of intrinsic disorder. As I started my academic laboratory, I was very interested in upstream signaling. The idea that, upstream of MYC, there was signal integration from surface to nuclear growth factor signals. But then what happened at nuclear chromatin was this sort of solid state transmission. And as MYC would bind to DNA, it would recruit a host of chromatin modifying enzymes to hyperacetylate local chromatin. And we hypothesized in my lab that BRD4 might be an integrator of that signaling, because BRD4 has an acetyl-lysine binding domain, actually two of them, at its amino terminus called a bromodomain, and at its carboxy domain, it can recruit PTFB or CDK9 to phosphorylate polymerase, releasing it from its proximal paused state, expressing the MYC-dependent genes. And so scientists at Mitsubishi Tanabe and ultimately our laboratory would innovate first bromodomain inhibitors, GlaxoSmithKline 2, including the chemical probe JQ1. And JQ1 is a fantastic molecule because it disrupts a protein-protein interaction, but we were afforded a greasy pocket to target. This acetyl-lysine recognition pocket in the bromodomain turns out to be so druggable, even a doctor like me could do it. And in BRD4 addicted cancers, we witnessed differentiation events, and true to our hypothesis, MYC is downregulated by BRD4 inhibition. But there were a series of assays that we imported or created in order to discover, optimize, and assess the differential activity of bromodomains. There was no kinome scan for bromodomains when we started. We learned about differential scanning fluorimetry from the Structural Genomics Consortium. And at Novartis, I've even seen this assay utilized for different protein folds in a high throughput format in a very incisive way. We brought in alpha screen technology, bead-based chemiluminescence from PerkinElmer, and this allowed for a very sensitive measurement with index weak ligands to be used all the way through lead optimization. And ultimately, with DiscoverRx, through a cold call to them, we did create a phage-based bromodomain lysate-based profiling assay called BromoScan. And so with having a taste now of inhibiting protein-protein interactions, but really needing to get beyond these greasy pockets, started to focus more and more on direct inhibition. And, I hate these lectures where all of the data is so clean and it looks like everything works. And so I thought I'd quickly tell a story of a project that didn't work, but led us, importantly, to consider a new path to a waterfall. So direct inhibition of a transcription factor and its target can be on the protein itself, or it can be on one of the vital members to which that protein binds. An inhibitor of MYC and MAX, as shown here, would be very exciting, but there's no overt hydrophobic invagination on this protein-protein complex. Whereas MYC engages any number of other targets directly or indirectly here from StringDB, and perhaps one could create an inhibitor, we'll say, to the trap protein that's vital for MYC-dependent coactivation. But first, Rick Young and I set out to make direct inhibitors of MYC and MAX. And this is a challenge because of intrinsic disorder. I mean, MYC has like 15,000 or more publications, but there's really no bona fide MYC inhibitor. And it's a challenge with MYC because, this lack of structure is characteristic until it ultimately finds MAX. And so admitting defeat that we wouldn't be able to necessarily bind MYC and MAX directly, we thought, could we lock MYC? Could we find MYC? Could we induce the fit with a small molecule to MYC that would lock MYC in a low energy state such as that it would not be available to MAX for binding? And so was the hypothesis of our work. Now, there are some pioneers of MYC drug discovery, and I want to acknowledge the high regard I have for Drs. Vogt, Chobnik, and Berg, who worked very hard studying thousands of commercial molecules in order to discover inhibitors of MYC-MAX heterodimerization. They were largely working with HLH, leucine, zipper minimal domains, but because we just wanted to lock MYC in a low energy state so it could never find MAX, we worked with full-length MYC. And our theory was, a little bit like what's old is new again, that since those seminal works were done, technologies had advanced in assay development as well. And perhaps with a super high sensitivity assay built around MYC, MAX, and EBOX DNA, we could find index chemical matter to subsequently advance. And so with support from the National Institutes of Health, and the Damon Runyon Cancer Research Foundation, we set up an alpha screen assay that locked EBOX DNA through streptavidin conjugation on a donor bead. And then MAX was affixed to an acceptor bead such that soluble MYC would be available to MAX in DNA, giving off bioluminescent light, or it would not be. It would be depleted by our small molecules. And we tuned the assay to exquisite sensitivity to MYC through dose ranging studies, and then optimized it for assay robustness and carried it in to pilot screens. Now, this was all at the Dana-Farber Cancer Institute, and where we didn't have access to large chemical libraries as we do at Novartis. But we didn't know what kind of molecule would inhibit MYC. And so we loaded up with a diverse chemistry as shown here on this slide, and in the fullness of time screened about 80,000 molecules. Now, many things hit in this sort of assay, but most of them are these so-called Payne's molecules that die in subsequent assay triage. But carrying hits into dose range study in alpha screen as a primary secondary assay, we found a natural product. We found this quinoxylene dione, aryl sulfonamide, and we found these dimethyl phenylpyrrole molecules like JJM288 shown on the right. And they had reasonable looking curves and low micromolar activities. Well, we got very excited about mitorubrinic acid. We have a colleague, John Porco, at Boston University across the river, who was very excited about this chemistry. And it's always great to have a chemist dedicated, knowledgeable about an assay positive. This is a natural product, and so not trivial to make, but John was terrific at that. And it retested not only in dose response, but it seemed to block MIC-MAX association, in the old school gel shift assays that I did during my training. It was not active in anti-proliferation assays in MIC-dependent cancer cell lines, but we thought that that was okay because we didn't expect that type of cellular activity right out of the gates. And so we took a series of derivatives of this molecule forward into subsequent secondary study. We learned that the carboxylic acid was really critical. That will become a theme more evident in a moment when I talk about the pyrols. We had access to about 12 structurally similar molecules and tried to create some sense of the structure-activity relationships. But really our inability to elaborate larger libraries and to pare down these molecules to small constituent pieces, and my own departure to Novartis interfered with the development of this series. In the next chemical series through SAR by catalog, we were able to get a lot more chemical equity in our hands. And you could see that in high single-digit micromolar potencies, we were able to block MIC-MAX in a gel shift assay, and lending some support to this thread. But most of the molecules we would access would prove to be wholly inactive, and there appeared to be a flat SAR, and flat SAR always really scares me. The next set of molecules we looked at were this pyrrole series, and there the index activity in gel shift wasn't really terrific, but it was selective for MIC and MAX over MAX and MAX. And for such a small molecule, we found that to be compelling. We worried that this might be a pan-assay interference molecule. And so I know you're all very familiar with this, but these aggregator molecules tend to lose their activity, as shown from the upper right red to blue shift, in the presence of higher concentrations of detergent in biochemical assays. And as best we could tell, the JJM molecule series did not seem to have that particular challenge. And so Jason Marino, then a postdoc in the laboratory, would create a series of molecules built off this simple scaffold, made about 400 molecules, tested them all at dose response. And we learned a little bit about sites of substitution that improve potency, vital functionalities like, unfortunately, the carboxylic acid, and other places where, as would be, should we get structural information, we knew were permissive for substitution. But the acid turned out to be a real problem, like for the development of phosphatase inhibitors for so many years. We just couldn't get away from this negative charge, which was really limiting our cellular activity and our ability to really test whether MIC and MAX inhibition could occur in a cellular context. And so we worked through a number of different pro-drugging strategies inspired by the development of antivirals so many years ago, and ultimately found AM esters and other derivatives that would lead to some demonstrable cellular activity. A series of compounds were realized that had single-digit inhibition of MIC and MAX in vitro, and started to have some characteristic MIC-like behaviors in cellular assays. One litmus test that I've always really appreciated in our own lab science, and often as a reviewer I asked for, is to try to ask the cell what state it's in using the measurement of multiplexed RNA detection. And now, honestly, with the availability of nascent RNA detection, GrowSeq, I just read a brilliant paper from Phil Sharp's laboratory on single-cell GrowSeq. I think these will be fantastic measurement tools to assess whether a small molecule in a cellular context is having a target or a pathway-specific behavior. And so we created what we call the Uber-MIC list. These are open reading frames that have high amounts of MIC at their promoters, and that with perturbation of MIC, this was before we had the DTAG assay, but with perturbation of MIC levels with doxorubicin induction or repression, I should say doxycycline induction or repression, we could witness dynamic immediate changes to MIC. And with this Uber-MIC list of genes upregulated, like P21, and genes downregulated, as shown here, we took these molecules through their paces to assess whether they had MIC-specific behaviors by transcriptional profiling, by patterns of cell sensitivity to MIC addiction, and even phenotypic suggestion of MIC-like activity in neuroblastoma cells that without MYC undergo phenotypic changes. A number of molecules were made by Jason. He really did a heroic job, and we could never really get below 1 micromolar for these compounds. Here's an advanced molecule that had pretty excellent selectivity for MYC and MAX over MAX and MAX. It did lead to growth arrest, but importantly, not apoptosis in MYC-addicted cell lines, as one might expect. We handed this project over to Syros Pharmaceuticals that Rick and I started. And when I went to Novartis, I stopped getting those emails. So I hope that they're still working on this science. We didn't create a MYC inhibitor, and this is on my bucket list for my time in science. I just work with anybody who is close to having one. But there are other ways to approach MYC protein-protein interactions, and this we did pursue quite intently at Novartis, and developing inhibitors for the interaction of MYC with some of its effector proteins. And here recently published is work from our group in chemical biology and therapeutics to realize first small molecule inhibitors of the MYC interaction with the WDR5, WD40 domain. We created at NIBR the first inhibitors of the WD40 domain by targeting the EED methyl lysine recognition motif on the polycomb repressive complex 2. And this really deorphanized the whole of the WD repeat domains for therapeutic discovery. And so Dr. Ding and colleagues at NIBR led an effort to create small molecules that are potent and selective engagers of WDR5 and do indeed compete MYC away from that transcriptional effector protein that recruits downstream chromatin complexes. I think that protein-protein interactions in and around transcription actually turn out to have more pockets than I initially thought, being a MYC guy for so many years. Also at NIBR, we had great success in developing the first of their kind inhibitors of the TEAD-YAP protein-protein interaction. Now, you must know that YAP-TEAD is a downstream effector of the Hippo pathway, very important for organogenesis. It's been intently studied in cancer cell biology as a pathway of addiction for cancers like mesothelioma and many sarcomas. But no YAP-TEAD pathway inhibitors had been realized, though some that activate YAP-TEAD, like LAT's antagonist had. And so we undertook to map the protein-protein interaction space functionally and biophysically between YAP and TEAD. TEAD is showed here in gray, and you can see the way that there's alpha helix engages this sort of long open channel on TEAD. But then there's this very interesting sort of omega loop that binds into a side pocket. And a very detailed biophysical dissection established that this omega loop actually contributes more to the binding event. And there have been so many successful examples now of targeting alpha helical interfaces interactions, but keying in on a tryptophan off of this omega loop that inserted itself into a deep-ish pocket into TEAD, we launched a structure-based drug discovery campaign building from tryptophan-inspired small chemical matter led by Jose Duca and colleagues in computer-assisted drug discovery, ultimately found stereo-specific fragments that would engage TEAD, from which we built out the first chemical tools to study the TEAD protein and YAP transcription factor displacement. These molecules are now advancing into human clinical investigation, and man, it got really hot. Actually, not at this protein-protein interaction site, but at an allosteric site involved in perhaps S-auto pulmonylation. 3 groups, actually now 4 groups, because Genentech has just published a brilliant paper this week, realized small molecules that engage this auto pulmonylation site. And so many of them leverage electrophilic covalent chemistry to engage a cysteine residue, one of several cysteine residues in that region. And I would just highlight that we have really exploited the concept of mapping electrophile library interactions with proteins of interest using exactly this approach of MS-guided covalent tethering to identify covalent fragments, and then with structure-based drug discovery, move through to potent and drug-like molecules. This is a smattering of data from Nathaniel Gray's brilliant paper on YAP-TEAD inhibition, where lo and behold, blocking S-autopalmitylation, or at least the site believed to be responsible for it, there's an allosteric modulation of TEAD structure that causes all YAP isoforms not to bind. And so I think it's going to be very exciting to watch direct TEAD antagonists as well as these allosteric modulas, or both in combination, Dare to Dream, used in patients with mesothelioma, and we're just so hopeful. All right, now in the 20 to 25 minutes remaining, I want to reflect on the science of protein stability, which is so exciting right now. And I really believe that this is the new path to the waterfall for these transcription factors, even those that exhibit real intrinsic disorder. We've worked hard on those projects. Each of them took about 5 minutes to talk about, but they're 7 to 10 years of science. But we came upon, through the study of the pharmacology, believe it or not, of thalidomide, a new capacity to target proteins that are otherwise intractable in small molecule drug discovery. Now, I bet a few of these slides you'll be familiar with from prior engagements at the NIH, but I'm just going to try to set the stage through the lens of assay development. Phenotypic screening and target ID remains, I think, especially in today's era of SciLac mass spec, just an incredible opportunity to understand pathway biology through the lens of small molecules and really expedite this sort of holy trinity of the small molecule, its biophysical protein target or targets, in the case of molecular glues, and a phenotype of interest. For many years, as a myeloma physician, I prescribed the IMiD molecules for patients, not knowing what they mechanistically, pharmacologically do to those cells. Ralph Mazicek and I performed ligand affinity chromatography with derivatives of Revlimid. In our case, we did an aryl amide coupled Revlimid, and these studies were densely negative. We failed to pull down proteins with affinity chromatography using Revlimid in the presence of or absence of a soluble competitor. And this was very disappointing. But then Hiroshi Hondo's paper came out a year later, and it was very exciting. See, Hiroshi made this aryl ether, and just that substitution turns out to facilitate the recruitment of the cereblon DDB1 ubiquitin ligase complex, which I found very exciting, because if Hiroshi could pull ubiquitin ligase complex onto a polystyrene bead, potentially we could direct it around cells, as the popular name PROTAC suggests, to other proteins of interest. The rationale for targeted protein degradation is well established, and I'm sure very well appreciated by this group at NCATS. But to be honest with you, though there's been an explosion of this field, the principal use case for protein degradation has been bifunctionals. And again, I think there could be some fantastic medicines that come out of that, but this chemistry used just for bifunctionals really only helps you to degrade things that you can already bind to. And so when I got to NIBR, into this wonderful environment of just unrestricted intellectual capital, we tried to swim out to the next breaking wave. And in 2016, we started an effort to make molecular glues for therapeutic discovery. Now, we had a lot of knowledge from the work of my lab and others, and I'll just flip through this because this is multiply published. For sure, engaging the cereblon ubiquitin ligase with a tether, and in this case, a BRD4 binding small molecule, could facilitate protein association and then witness the expected hook effect. And leveraging this highly evolved and selective protein machine, we could achieve unprecedented pharmacologic selectivity of protein degradation. Indeed, these molecules even worked in vivo. And in this manuscript, we solved a lot of the challenges of the field, but the reviewers really wanted to see more. They wanted to see this extended to diverse targets, and we really wanted to see it extended to targets that are unligandable. This is one slide to summarize a lot of progress in the field. It's quite selective for the proteins. In fact, sometimes, as shown in this middle panel, you can take a ligand that's not so selective, like this BRD7/9 inhibitor, and degradation proves to be even more selective than the ligand itself. And that surely has a lot to do with the geometric requirements to convert induced proximity to functional degradation. We learned that through linkerology and sampling different chemical matter on both sides of the interface with the PROTAC technology, we could, as shown here with Eric Fisher's laboratory, engender real selectivity even within the bromodomain family with non-selective BRD2, 3, and 4 inhibitors. We published, and others have subsequently validated this, that these molecules are like little catalysts that degrade the protein, but they themselves don't get degraded. Here, a substoichiometric shift in a cell line that has the cereblon ligase or lacks the cereblon ligase, and you can see how viability is impaired. We and others have shown that degradation and inhibition are just not the same. In some cases, better, and in some cases, like BRD4 degradation, worse, where BRD4 inhibition downregulates a small number of genes, BRD4 degradation downregulates all of productive transcription elongation. And that really taught us something very fundamental about the role of BET bromodomains as master regulators of proximal promoter pause release and transcription elongation, now well characterized by Ali Shalatafard and others. But really interestingly, I think, and not a lot of people have done much with this, so I'll just mention it here. Sometimes in drug discovery, we drug the wrong thing. You drug the bromodomain of TRIM24 because you can, but then the bromodomain inhibitor doesn't do anything. And when we took these inactive ligands and we turned them into active degraders, we could really witness phenotypes associated with CRISPR target validation studies. Here, the arrested proliferation of leukemia cells. So the field's just on fire. And on the heels of this and other papers, people started putting this happy little phthalimide, on all kinds of molecules. And it's been really exciting to watch that science mature, and I think really energizing to see so many organizations building out capabilities around targeted protein degradation. There are a lot of challenges, I believe, and I've listed a few of them here. I won't read them to you, but leave it up just long enough that you can see them. The big ones I want to talk about today is this requirement for ligandability that I mentioned. I think it could be very interesting to have an AR degrader that does a better job of degrading nuclear androgen receptors or estrogen receptors. And I think that maybe even some of these molecules could degrade mutant forms of the estrogen receptor and androgen receptor, and they could be important therapeutics. But we've got to swim out to this next breaking wave and get this chemistry to unligandable targets. And that's where I want to close by just sharing a reflection on these molecular glues. So I don't know that this is a real term of trade, but we, in the parlance of Novartis, talk about these molecular glues ever since the pioneering work at Novartis around the characterization of the mechanism of cyclosporin and FK506. And as you know, that glues can come in two flavors: intermolecular glues, which I'll talk about today, that create protein-protein associations that wouldn't otherwise exist. They bind into proteins and then create a complementary interface, a neomorphic interface for a protein that wouldn't normally engage, like short-circuiting a signaling pathway. But there also can be intramolecular glues, and I think this is where the action is. These are molecules that bind to multiple domains in a protein, restricting it into an inactive conformation. I encourage you to learn about our work to create the first SHP2 tyrosine phosphatase inhibitors, as well as the first clinical stage inhibitors of the NLRP3 inflammasome, both of which function as intramolecular glues. The glue-like activity of the phthalamides would ultimately be discovered by Bill Kalin working with our group and Ben Ebert across Longwood Avenue simultaneously. And this was pretty cool and unexpected. The thought was, even Hiroshi thought, I thought that the phthalamides would bind cereblon and then inhibit its capacity to degrade, inhibit its E3 ligase function. And there might be then proteins that would be stabilized by the phthalamides. And Bill had a really cool assay that allowed for the direct measurement of a gene of interest relative to a luciferase control. approached me to say, what might we try in this assay? And I thought, well, let's see what these thalidomide drugs are stabilizing. And lo and behold, they were actually depleting Ikaros 1 and Ikaros 3. And that's when it got really interesting. Independently, Ben Ebert and colleagues at the Broad Institute took a proteomic approach and ultimately found that Ikaros 1 and 3 were degraded in cells treated with IMiD molecules. And Eric Fisher would subsequently show that another transcription factor called SAL4 is degraded in iPS cells. We were working in cultivated adherent cells, but that in iPS cells, another transcription factor, SAL4, would be degraded, and that this probably explains the phocomelia associated with thalidomide. Nico Tomei at Novartis, our Friedrich Miescher Institute, and Phil Chamberlain at Celgene, where these IMiDs were pharmacologically brought to market, therapeutically brought to market, would both, again, simultaneously report that the mode of molecular recognition of these phthalimide molecules is for this glutaramide warhead to go into a tri-trip cage, really extruding water, and through strong hydrophobic interactions, bind tightly into this cereblon pore, and then protruding out this little bit of aryl matter would create a neomorphic interface for proteins to bind. Now, it's interesting, at Novartis and also in the published literature, characterizing the transcription factors that are degraded by these small organic molecules, very subtle changes to the permissive exit vectors will have a great influence on what proteins are degraded. Unliganded, there's no recruitment. We're still trying to figure out what cereblon does. Lenalidomide will degrade 3 proteins, Ikaros 1 and 3, as well as casein kinase 1 alpha, very important for myelodysplastic syndrome with 5q minus. Some won't do that. And others, very subtly differentiated in this attachment chemistry, will degrade other proteins like GSPT1. In fact, of the 19,500 proteins in the cell, there's probably 6,500 that have, structural data. And 2,000 of them have this motif that we've come to recognize as being the motif that phthalimide recruitment can bring to cereblon. And it's a little beta hairpin turn, just a little beta hairpin turn that ultimately binds into this interface. And that beta hairpin turn is often, but not exclusively, positioned in the context of a zinc finger. And so if you look for just the zinc fingers, you get about 2,000 proteins. But with the advent of AlphaFold, recognized yesterday by the Lasker Prize, by AlphaFold, you actually see this beta turn in almost 3 times as many more proteins. And so, if all we do is build on the pharmacology of the phthalamides, there are a lot of proteins that we might create first direct acting and selective degraders of. And so at NIBR, again, in about 2016 fall, we undertook to elaborate a library of cereblon biased small molecules for target-specific and phenotypic drug discovery. And this is the chemical space that was very thoughtfully mapped out and ultimately realized through CombiChem. In order to test these molecules, we needed a suite of assays, some that were quite holistic, just show us what's degraded, a Go Big Proteomics project. There were others where in this list of proteins broadcast out to the NIBR community, we asked, which of these proteins could you get excited about, is relevant to the disease you study? And receiving input back from the organization, we set up target-specific screens with this chemical library and really empowered phenotypic drug discovery at all 6 of our sites with this capacity. So bioinformatics was a big part of this, degradation proteomics as well. But I want to highlight an assay platform that really proved very powerful that you may be less familiar with, which is the Orionis so-called AlloGlue platform. I met these Orionis guys at a cocktail party in Boston thrown by some venture capitalists. And I got to admit, I was rolling my eyes going to this thing. But sitting there and talking to Nico and colleagues from Orionis, it seemed that they had built something very exciting. These brilliant molecular biologists had created an assay system that could, in cells, using relatively conceptually simple but experimentally elegant two hybrid approaches, they could create bioluminescent signals when proteins would engage one another, theoretically for protein-protein inhibition. John Tallarico and I got excited about using these for protein-protein induced proximity or induction of protein-protein binding for molecular glues, and worked together between our institutions to optimize their platform to detect very subtle and low energy interactions from which we could build through lead optimization in these assays, potent molecules. And as Bill Forrester presented at the New York Academy of Sciences, so I can share with you here, these cereblon biased assays would have cereblon be one of the receptors, and then any number of zinc finger possessing proteins or beta term possessing proteins would be tuned for the capacity to create this signal. And what we found was actually a very large dynamic range once fully optimized and miniaturized to 384 well plate format. Nico has kindly shared these slides, and I would like to share that these guys are really brilliant. And since this collaboration to create these cereblon biased glue assays, they've really run with this concept in an exciting way, and are now able to identify allosteric modulators that induce this. They've had a chance to study conditional glues that facilitate protein-protein binding in the presence or absence of a cofactor. And you could imagine that for heterobifunctionals, they work just right out of the box. We really focused on a ligase-centric process in the very beginning. With a library bias for cereblon, we could whip through a large number of potential binding partners against another axis of a large number of small molecules. But it's not lost on all of us that there's also a target-centric approach where if you care about a target, say MYC, you could then use a large library of compounds against a large library of ligases and look for potential interactions in that manner. There is nothing TPD specific about this platform. This was just its first exemplification. And I found it very exciting that 3 years after we initiated our collaboration, that others here, Genentech, would see the same value in the Orionis platform, and delighted to see its broader use beyond the TPD and induced proximity use cases at NIBR. I just want to show you a little bit about what this data ultimately creates is really a lookup table of various targets and compounds, some of which exhibit exquisite selectivity, others of which exhibit real potency, and then might be tuned for subsequent target-specific potency. We found a lot of really strong hits. I will show you what these targets are in a minute. Using like really strong biophysics and protein structural biology, we could weed through the imposter molecules pretty quickly and get to molecules of real interest. And with these capabilities, we would ultimately realize the first Helios protein degrader. Helios is a cancer target, but it's actually not in the cancer cell. It's in the T regulatory cell. One could deplete T regulatory cells, potentially releasing the suppressive effect on T effector cells, but there would be real autoimmunity associated. And so we thought, could we stun the T regulatory cell state such that, in a reversible manner, such that we could release inhibition on the T effector cell and augment the capacity of checkpoint inhibitors. And indeed, through molecular, biological, genetic, and chemical genetic methods, this would prove feasible. And so Helios is also called Icaros 2, and you can see that all the Icaros family members have this common CXXCG, but they do bear distinct appending sequences. And none of the IMiDs that are today pharmaceutically used degrade Helios, which would have been nice. We could go right to combination studies. But we did find that pomalidomide had a weak capacity to engender a protein-protein association here with the HYBID assay that I'll show you in a moment. Actually, I'll show you right now. So in the beginning, we don't look for degraders, we look for glues. And then we subsequently confirm that work through biophysics. and then take them through a series of biochemical ternary assays, and ultimately through lead optimization, endow them with degrading properties. And a workhorse of this assay cascade is the HYBID assay, ingeniously developed by Promega as a bioluminescent split enzyme reporter system. You surely know nanoluciferase as a 10-stranded beta barrel. At this red arrow is the spot of dissection of this enzyme, which can then be recapitulated as two distinct split enzymes here over a variety of concentrations and different peptides to recreate an enzymatically competent enzyme. In their index paper, they nicely show that rapamycin can assemble FRB with this 11S large domain or large bit that has FKBP fused to various peptides and can show BRAF-CRAF association in the presence of small molecules as well. This assay is not only good for protein-protein association, but also to monitor protein stability, as Danette Daniels has published in a very elegant system where she drop ships the HYBID right into a gene of interest, and then on plasmids has the large bit delivered that can allow for bioluminescent measurement of just one protein stability in a real-time high-throughput assay, giving insights into the kinetics of protein stability. And from this, we're able to gather the metrics to advance molecules through to clinical candidate, which is the Dmax, the degree of degradation, the time degraded, as well as, not shown here, the DC50. These assays performed a different way also allow you to witness the capacity of a molecule to engage its protein target. Very important when you're optimizing the biophysical properties of bifunctional molecules to know what their cellular access to target really is. Well, there's a lot of work in one slide, but we ultimately would take these insights from pomalidomide and use that core and then start to very carefully build out towards engendering more Helios protein-protein association, and then ultimately protein degradation. Shown here is DKY709, which is a clinical stage Helios degrader being studied in solid tumors presently, and one can see that it exhibits strong degradation capacity at submicromolar concentrations. We solved the high-resolution crystal structure of DKY, recruiting the Ikaros zinc finger 2, as shown here, and one can see the beta turn and glycine of Ikaros 2 adopting the structural recognition event as expected, and the way in which DKY bends itself around this carboxylic acid on cereblon to permit engagement from Helios. This molecule sailed through preclinical toxicology studies that exhibited potent and durable degradation of Helios in primates, because this chemistry is hard to do in mice owing to species differences in cereblon. This molecule was studied in human T regulatory cells and potently degrades Ikaros 2. And in functional assays of vaccination and T effector cell proliferation, these molecules will be comparably active. I'm excited to share that this molecule today is in human clinical investigation, and as presented with human exposure, drops Helios levels in FOXP3 positive T cells. So in closing, molecular glues are really pretty exciting. As we get more ligands for new ligases, each of these, as for the glutaramide containing phthalamides, become an opportunity for building out focused libraries and assessing protein-protein interaction and ultimately protein degradation. And I do believe that for transcription factors, soon to be presented over the next few years, there's just a big opportunity to leverage this, even just this chemistry, for a broad therapeutic benefit. I only talked about the cereblon glues here today, but at NIBR we, I believe, have about 8 clinical stage or near clinical stage molecular glues, everything from glues that modulate spliceosome function to, as I mentioned, intramolecular glues of big cellular machines. It's all a little bit what's old is new again. I want to credit Stu Schreiber and John Clardy and Chris Walsh, so many others that really thought about modularity in small molecules, Jerry Crabtree, But creating these ligand-efficient molecular glues really avails an opportunity for drug discovery. I know we have a few more minutes for questions, and thank you very much for the invitation to join today.
Thank you very much, Jay, for a wonderful presentation. So yeah, so there's a few questions, so we'll just address them. We have a few minutes left. So we'll start with the first one. So this is from Matt Hall, the director here at the Early Translation Branch. He says, thanks so much for your presentation, Jay. The observation that a BRD4 degrader has a greater impact on downstream transcription than an inhibitor is intriguing. So much target discovery is via CRISPR that deletes a protein. Then drug development pursues an inhibitor that doesn't have the same effect. Are we moving, the question is, there's two questions. So one is, are we moving towards a work here where degraders become the preferred modality rather than inhibitors? That's question one. And the following question is, or a world where we need an inhibitor and a degrader to decide which approach is best early in a drug discovery program? Appreciate your insight.
Well, thanks. And Matt, great to be here with you. I don't think we're moving to a world of degraders. You know, we have to be very careful when the pendulum swings towards these new areas that, you know, we don't go running with a big basket to collect these strawberries, you know. And I know you're not the kind of scientist to fall prey to that. It is true that degraders are a very nice follow-on to discoveries from CRISPR, but using tiling CRISPR and using a knockdown or knockout and add back strategy, you know, we can really dissect what biophysical capacity a given protein has. If I found a kinase, multi-domain kinase that was interesting in an inflammatory disease, I wouldn't rush to degrade it because it's so readily inhibited. And degraders, by the way, come with real challenges as well. In cancer, as Konstantin Mitsiadis, as Georg Winter have published, there's just so many ways to evade a cancer degrader. I mean, the whole of the ubiquitin ligase pathway can be downregulated, epigenetically silenced, knocked out, selected against, and degrader activity is lost. So as excited as I am about degraders, if given the choice, I'd make an inhibitor.
Thank you so much, Jay, for that insight. All right, more questions coming in. So thanks for a great presentation. I would like to know if there are differences in sensitivity between NMYC and LMYC. And also the same person asks another question, which is, have you tested these compounds in cancer cells with MYC gene translocations?
Well, I'll take the second question first. Yes, some of these cells are Burkitt's lymphoma cells that exhibit a very high degree of MYC and real MYC sensitivity. Some of these are neuroblastoma cells that have NMYC or CMYC characteristic dependence. We did not ever really get a cell line that we could be convinced was LMYC addicted. This work predated, however, these grand experiments like the DepMap, that perhaps today you could find something LMYC sensitive but not CMYC sensitive, or engineer it to be as such, as so much of this work was done contemporaneous with the index discovery of CRISPR. But I think it would be very important to have, to do this work again, to follow up on this chemistry, to have a paired cell line system, the exact same chromatin, but with LMYC, NMYC, CMYC all represented. And we did not have those tools at that time as easy as they would be to generate today.
Thank you, Jay. I appreciate it. Two more questions. We're right on time. Do you mind if we take two more questions?
Go ahead.
Thank you. So are gene control medicines somewhat unique in terms of pharmacodynamics? Basically, is there any benefit such as long-lasting memory post-drug exposure as compared to conventional small molecules that target signaling oncogenes in parentheses, where signaling rebound and adaptive resistance often happen very quickly?
Oh, Michelle, I love this question. I'm racking my brain to think of one, but it surely could happen. It could happen for a transcription factor. I'll give you an example. Notch inhibition. When one genetically inhibits Notch pathway, or even through gamma secretase inhibition of Notch processing at the plasma membrane, a binary cell fate decision is taken. And that cell becomes, you know, a goblet cell of the gut, and it never goes back, even if you wash the drug away. a H3K79 dimethyl methyltransferase inhibitor to DOT1L leads to irrevocable differentiation of a pediatric leukemia. And I believe the same is true for menin inhibitors. And so I think the answer is yeah, but signaling pathway inhibitors do that too, like Forbol ester in so many circumstances. So I think that the answer is not always, sometimes, but not exclusively.
Thank you so much, Jay. One more question. And then Jacob Rose says, fantastic review and talk. I was wondering if you could elaborate at all about your explorations into glues targeting splicing components, as you mentioned, as you just mentioned. Thanks.
Yeah, well, let's just get together in the conference room next door. What I can say is that a phenotypic screen led by our neuroscience group that's published in Nature Chemical Biology, very elegant system, and a cell-based system looking for molecules that would alter splicing of SMN2 so as to compensate for SMN1 loss. And then the follow-up assays were equally exquisite. And in this cell-based assay, we thought we must have found some protein modulator of splicing, and a lot of work was done. Chemical pull downs with ligand affinity chromatography, photo cross-linking and labeling, and no protein was discovered. Ultimately, it was thinking that maybe it was RNA being targeted that led to the realization that this is engaging this the U1 SNRP, the spliceosome, and this pre-processed piece of RNA, not mRNA. And the biophysical characterization of that interaction was extraordinary and subsequently validated by Pete Schultz and colleagues. So I think it's real. These molecules went into human clinical investigation, and we could even see SMN2 stabilization in SMN1 null patients. And so on the heels of that, we and others have looked for other molecules that modulate splicing, and there's a goodly number of them, but they're not terribly selective or as sequence specific as what we saw with that program. But I do think that, if you could drug RNA, then nothing's undruggable. So it'll be very exciting to take that work more seriously.
Fantastic. And Michele is following up here, and he is saying, how can we access the NIBR Broad Open Bell platform? And there's a funny question afterward. Can we get a free slot for asking questions in this chat?
Yeah, since I left Novartis last November, I'm wondering the same thing. It's an amazing resource. It's not an unlimited resource. And I think that the corresponding author, Corinne Briner is off to Genentech. I'm off to a next chapter I can't talk about just yet. And Stu Schreiber remains at the Broad Institute, and Fred Zekri at Novartis. And so I think reaching out to those corresponding authors, Fred Zekri and Stuart Schreiber, and just asking for, under what circumstances and for what project could this library be utilized. We had hoped that this could be a pathway to taking a beautiful chemistry, this natural product like structurally diverse, three-dimensionally complex chemistry, DOS chemistry, that sometimes can be a little hydrophobic, a little greasy, but put it on DNA as the ultimate solubilizing vehicle. And then in finding interactions through exemplar members of this library, using cheminformatics, you could even drill down to what the likely structural determinants of binding are, facilitating follow-up chemistry in a very focused way. So please reach out to Fred Zekri at Novartis, or or Stuart Schreiber at the Broad, and perhaps they could tell you, and then you could tell me.
Fantastic. Thank you so much, Jay. This was a fantastic presentation. We appreciate the time that you gave for us today. Very thankful for your wonderful talk. Thank you, everyone, for joining, and you all have a wonderful day. Thank you so much.